Good afternoon, everyone. So today I'm going to talk about using Burkholderia species to control soil-borne pathogens. My name is Hongwei Liu. I'm from Western Sydney University. So firstly, I just briefly introduce about the project. This project aims to deliver innovative solutions for controlling crucial soil-borne or root-associated pathogens in horticulture crops, enhancing Australian agribusiness profitability and growth. It will develop bio-based technologies to utilize crop-optimized microbiomes, plants' immune systems, and biochemical products to revolutionize the way root disease are tackled in both protected and field conditions. We are planning to introduce unique tools to suppress disease and weed host, offering horticulture farmers additional income systems. So in this talk, I will use Fusarium pseudogrammaneum, the pathogen, as an example. It causes the corn rot disease of the wheat plant. In Australia, wheat is a major winter crop, which has an impressive annual production estimated around 25 million tons a year. Despite of this, the agriculture sector faces considerable challenges, most notably from the fungal pathogens like Fusarium pseudogrammaneum, causing estimated annual yield losses reaching to $468 million. Particularly alarming is corn rot disease caused by the soil-borne pathogen, Fusarium pseudogrammaneum, that poses a substantial threat to our cereal industry. Despite numerous attempts, currently there is no available commercialized methods to control this pathogen due to many obstacles. It is crucial that we can focus our research efforts on understanding complex interactions between the pathogen, our soil system, crops, and also how the interactions are influenced by the climate change scenario. Bacteria, integral to the ecosystem functionality and plant performance. It has great potential for controlling soil-borne diseases. As an alternative to traditional synthetic chemicals, bacteria present an environmental friendly, effective, and economic method for pathogen control. And in my study, we isolated the bacteria from wheat plants with the aim to identify effective strains to control the pathogen. As shown in the slide, we found we isolated multiple bacteria from the wheat, and one of which could significantly inhibit the pathogen Fusarium pseudogrammaneum, and we identified the beneficial bacterium using the whole genome sequencing and the 16S RNA single sequencing. We found it was identified as a Burkholderia ambiferia. It has about 1.27 micrometers length, and displaying very promising implications for the controlling the Fusarium pseudogrammaneum. Upon placing the genome of this bacterium into the context of a comprehensive bacterial database, we observe that the bacterium genome, which is about 7.4 megabase pairs, and it contains 7,361 genes, which is significantly larger than the other bacterium of the same species in the database. This suggests that our strain, Burkholderia ambiferia, has an enhanced capacity for protein production, potentially aiding its colonization on the wheat plant. This finding also aligns with existing knowledge in the bacterial genomics, which is, as we can see from the correlation graph, there's a strong positive correlation between the bacterium genome size and the number of coding genes they have, which indicates that the adaptive capacities of bacterium with larger genomes in the plant. As this depicted in this illustration, it demonstrates key mechanisms that bacterium can benefit the plants. Among which, the bacterium can combat the pathogen via production of siderophores, and also can directly promote the plant growth via solubilize the phosphate in the soil to make it more available for the plant to use so that they can grow better. Upon further exploration of the genetic functions of this bacterium, we found the Burkholderia ambiferia has the pathways for siderophore production. Subsequent testing on agar plates also confirmed its capacity to produce siderophores. And similarly, we also found it has the capacity to solubilize the phosphate on agar plates, suggesting that it also has a potential to promote the plant growth by directly adding nutrients availability to the wheat plant. So now we identified an effective bacterium strain and also a good plant disease model or system. Then we can investigate the colonization process and how it impacts wheat plant growth. Through inoculation, the bacterium in different doses, namely 1 or 3 doses, and the sequences, which was earlier or later than the pathogen, and under varied conditions, which was interrupted or non-interrupted microbiome, soil microbiome. We have examined several key points, and we found from the 2 graphs on the left shows biomass production of the wheat shoot, and the right panel showed the biomass production of the wheat root in a microcosm experiment. We found a higher bacterial doses resulted in the increased wheat biomass production, and suggesting the higher dose application of the bacterium probably also had better soil colonization. The presence of bacterium, it doesn't matter it's introduced simultaneously with the pathogen or earlier, could all greatly enhance the plant disease tolerances. Upon further examination of the plants treated with the bacterium and the pathogen, we discovered that the expression of the plant defense genes was also impacted by both types of the treatments, which were the either by the beneficial bacterium or by the pathogens. However, these findings are based on my preliminary analysis, and the enhanced, more detailed analysis in the future are expected to provide a deeper insight into the mechanistic interactions among the plant, the bacterium, and the pathogen. Additionally, we also, as shown in the right panel, we also quantify the pathogen load in the soil and the plant using qPCR analysis. Our initial results suggested a temporal pattern in the colonization of the pathogen in the plant. More definite conclusions will follow as we further analyze and validate these initial findings. And now I'm waiting for the amplicate and the metagenomics data to further analyze these processes. Overall, our findings to date highlight the potential of the beneficial bacterium Burkholderia amblyophilia in controlling the fungal pathogen Fusarium pseudograminarium in the wheat plant. That's my talk today. Thank you for your attention.